Right. Welcome, everyone. My name is Kara Kaikini. I am the Executive Director of the Maine State Breastfeeding Coalition, and I use my she, her pronouns. And I wanted to welcome you all to our third educational webinar of our 2023 season. Our next webinar will be on August 3rd during World Breastfeeding Week. And we will have Dr. Julia Fritz and Virginia Weil from Maine Medical Center speaking about understanding feeding challenges from a gastroenterologist and speech and language pathologist perspective. So stay tuned for registration info. If you don't want to be added to our mailing list, let me know. And if you do not want to be added, otherwise, I will go ahead and add you from your registration. For today, I am so excited to introduce our presenter and to hear her presentation. Jessica currently practices as a primary care pediatrician at Maine Medical Partners, South Portland Pediatrics. She completed medical school at the University of Rochester in 2009 and pediatric residency at Massachusetts General Hospital in 2012. Dr. Jessica Rosenthal is the breastfeeding champion for her practice and has a passion for supporting families in their breastfeeding journeys. She helped create and facilitate a breastfeeding peer support group at her previous practice in Massachusetts. Outside of work, she loves spending time with her husband and 2 children. Jessica also happens to be one of our board members. Thank you for being here today, Jessica. Welcome and take it away.
Thank you so much, Kara. And I also use she, her pronouns. I'm excited to be speaking about this topic. It is one that is near and dear to my heart, although I will give the disclaimer, this is not something that I have a specific research area interest in, but something that I'm very interested in learning about. And it was very interesting to put this presentation together. So I'll be speaking about breastfeeding and the human milk, breastfeeding and human milk and the microbiome. So objectives for today, first to do a brief overview, what is the microbiome and why does it matter? Second, to look at factors that affect the maternal and child microbiome and how do milk and feeding fit in. And then third, to look at the microbiome's effect on disease in children. So first off, a brief overview, what is the microbiome and why does it matter? So I'll look to a definition from the National Human Genome Research Institute, and this definition states that the microbiome is the community of microorganisms, which includes fungi, bacteria, viruses, and some other things that exist in a particular environment. Some definitions differentiate between microbiota, meaning the organisms, versus the microbiome, meaning the organisms' genomes. And so those, I've sort of used those interchangeably in this talk. In humans, the term microbiome is often used to describe the microorganisms that live in or on a particular part of the body, like the skin or the gastrointestinal tract. And these groups of microorganisms are dynamic. They change in response to a host of factors, including exercise, diet, medication, other exposures, and we'll go through that a little bit more. So to think about this a little bit more, again, the microbiome is a collection of all these different types of microbes that naturally live on our bodies and inside of us. The function of the microbiome is to protect against pathogens, help our immune system develop, and enable us to digest food and produce energy. The impact of the microbiome, some of these microbes can alter the environment to increase toxicity, and others can sort of buffer and make these environmental substances less harmful. There are a plethora of microbes in the body. There are as many microbes as there are cells in the human body. The microbiome is very diverse. Each person's individual microbiome is different, and each body site within a particular individual also has a different microbiome. And it's a very dynamic sort of organ, if we want to think about it that way. It's the core microbiome is formed in the first years of life, but it can change over time in response to a number of different factors. So are microbes good or bad? We used to consider bacteria in particular that colonizes humans as bad, that these were pathogens that needed to be destroyed or treated with antibiotics. We now recognize that there are many good microbes. We sometimes call these commensal microbes, things that live with us in a sort of symbiotic relationship. And they can be helpful in many ways. For example, gut bacteria can help in the digestion of food. Skin bacteria can help break down lipids, fats to produce natural moisturizing factors, and microbes can provide colonization that helps with resistance of sort of bad actors. And just to put this in context a little bit, there's the idea of the genome and the microbiome. And so in 1953, the DNA double helix was identified by Watson and Crick. And fast forward to 1990, the Human Genome Project took off. And only 11 years later, the human genome was mapped, all 23,000 genes. And there was an expectation that this might explain human disease. If we understood the genes that are responsible for diseases, we should really understand, why people get diseases. And it's not that simple. And so in 2008, the Human Microbiome Project was commissioned, and this allowed resources to be allocated to better characterize the human microbiome and its role in health and disease. And what we've really started to understand is that there's this multi-biome, this interplay between our genome and our genetic predisposition, and then also our microbiome. And it's really these things working together that impact disease and later health outcomes. So a term that's really important to kind of familiarize ourselves with is dysbiosis, which is the disruption of the healthy microbiome. So the microbiome consists of all different types of microbes, as we've discussed. Some are helpful and some are potentially harmful. And this is a mostly symbiotic relationship where both the human body and the microbes themselves benefit. And then there are fewer, but some sort of pathogenic microbes that are more involved in promoting disease. And so in a healthy body, there's this coexistence of the pathogens and the symbiotic microbes that exist without difficulty. But if the balance is disturbed, and this could be by a number of different factors, such as an infection, certain types of diets, prolonged use of antibiotics, other medications, then we get dysbiosis, which is sort of a breakdown of this normal interaction, this normal interplay. And this is when the body can become more susceptible to certain diseases. So moving on to our second objective, and this is a big one, is sort of what are the factors that affect the maternal and child microbiome, and how do milk and feeding fit in? So the first thing to think about is there's this idea of the priority effect hypothesis. And this is this idea that the infant gut, the microbiome of the infant gut, which is a key actor in many, many functions, depends on the type and the order in which bacteria colonizes the intestinal tract. So sort of it matters what bacteria gets there first. And how does this happen? So newborns are typically inoculated with maternal, vaginal, and fecal bacteria during vaginal delivery. And we'll get into this a little bit more, but with C-section delivery, newborns are often colonized more with skin flora from mothers rather than the vaginal and fecal microbes. And then the second way that infants develop this microbiome is from human milk. The human milk microbiome is the second integral source of microbes for infants after the birth canal. And just to put in perspective, every day a breastfed infant ingests somewhere between 100,000 and 10 million bacteria, and 30% of the infant's bacteria come through the breast milk. There's also further transmission of the infant microbiome through contact with maternal body sites during oral and skin-to-skin contact, with other transmission from other household members, from even objects in the home, and also from other people and objects even in the hospital or the birth environment. So this idea that there are microbes in breast milk is something that hasn't always been understood. Initially, the idea was, it was thought that breast milk was sterile. In the 1950s was the first time that there was an observation that there are microbes in milk. And this was found when studying the question of, can infections be transmitted from to infants from their mothers through breastfeeding? And it was observed that there were microbes in milk. And then in the early 2000s, there was the first study that showed bacteria, and in particular, a lactobacillus type of bacteria, was found in the breast milk and then the stool of mother-infant dyads. And this was the first evidence that breast milk is a potential source of colonization of the infant microbiome. And so our current understanding is that breast milk definitely does contain microbes. And so what does breast milk do and how does it sort of contribute to the infant microbiome? So first off, there's direct seeding, meaning that the bacteria that is in the breast milk, studies have found that that bacteria does end up in the infant's microbiome in the gut. And then there's also indirect impacts. So breast milk also contains other compounds, prebiotics, other parts of the immune system, like immunoglobulin A in particular, and other compounds that also affect the colonization pattern of the infant's microbiome. And so early microbial colonization of this mucus tissue, these mucous membranes, especially in the gut, has a really huge impact on the immune system. It shapes the infant gut and the respiratory microbiomes. And there's this idea of this gut-lung axis because there's a lot of interplay between the gut and the lung. It supplies nutrients for bacterial growth and also other metabolites that impact bacterial growth. And so early intestinal dysbiosis, so the idea of the sort of we've lost the symbiotic relationship and this healthy balance in the intestine is understood to be a risk factor for later disease, things such as obesity, asthma, necrotizing enterocolitis. So the human milk biome contains tons of different species, in particular with bacteria. There are over 800 different types of bacterial species that are found in the human milk microbiome. Staph and strep and lactobacillus are some that come up frequently, but there are a number of different categories. And it's interesting to think about, how does this actually happen? What is the mechanism of colonization of the human milk microbiome? And there are two primary pathways. There's the enteromammary pathway, and then the idea of retrograde flow, and we'll go through these. So first, the enteromammary mechanism is the idea that there is bacteria in the maternal gut, and this bacteria translocates through the wall of the intestine. And so some of the bacteria from the mother's intestine and the mother's microbiome comes through into the lymphatic circulation. And that eventually goes to the mammary glands and ducts and ends up in the mother's breast tissue and then in human milk. And there's some studies that have shown that there's some physiological and hormonal variations during late pregnancy that cause more permeability of the lining of the intestine and allow for more of this translocation of bacteria and different components of the maternal microbiome to allow it to get into the maternal breast tissue. There are some specific systems are in place that help transport bacteria from the mother's mucosal tissues, especially in the intestine, to the lactating mammary gland of the breast. And to highlight this, and some of the evidence that shows that this happens is that there are bacterial communities that are found in colostrum that's been collected prior to the infant ever nursing. So it's there before the infant even touches the breast. The second pathway is the retrograde flow pathway. And this is a pathway that is from the infant's oral cavity into the mammary duct during feeding. And there are ultrasound studies that have shown this backflow of milk from the infant oral cavity due to the process of infant suckling. And so there's this bidirectional exchange of microorganisms between the breast and the mouth of the infant. And this can explain how bacteria that's commonly found in the infant's oral cavity or the human vagina, which the infant is typically colonized with early because of passing through the birth canal, can then also be found in human milk. There's also bacteria from the maternal skin that enters into the breast milk through the process of infant suckling. And also interestingly, just to note, the mammary tissue of the breast has its own microbiome that's different than the skin. And there, in 2014, there was a discovery that there is bacteria in the breast tissue of women both with or without a history of lactation, and also without any history of infection, that there's still bacteria there. And this is likely from the 6 to 8 breast ducts that have an opening on the surface of the human nipple that allows microbes to get in from the environment and get into the ductal system of the breast. And these same microbes likely influence the microbes of the human milk. So I've mentioned bacteria, but bacteria is not the only component of the human milk microbiome or of any microbiome. But bacteria is one component. And so when we are talking about the bacterial component of the human milk microbiome, that's the bacteriome of the human milk microbiome. Breast milk contains, like I said before, several hundred different bacterial species, and strep and staph are the most common. And these are common bacteria that are found on the skin. And this is universally true that strep and staph are the most common. There's no change with geography or other factors. And then a number of other types of bacteria are also seen, including bifidobacterium, lactobacillus. I'm not going to go through all of them, but these are some of the common types of bacteria that are found in the human milk microbiome. And interestingly, the overall bacterial diversity changes throughout breastfeeding and actually becomes less diverse throughout the course of breastfeeding. So mature milk is found to be less diverse compared to the number of different types of bacteria that are found in colostrum and transitional milk. And the composition of the bacteria changes throughout the course of breastfeeding, from skin and enteric organisms early on to more of the infant oral and skin organisms later on. Another component of the human milk microbiome is the virome. So this is sort of the viral part of the microbiome. And the type of viruses that form the majority of the human milk virome and the virome in general are these bacteriophages. And there's a picture of one shown here. And this is, they're called phages, bacteriophages. They are actually viruses that infect bacteria. And they can have different effects on bacteria. They can kill bacteria, and they can also provide some potentially beneficial genes and change some of the character of the bacteria. So newborns are initially colonized primarily by bacteriophages. Then these replicate in the infant's intestine, and this sort of has an impact on the particular bacterial growth in the infant's intestine. And so it has a really large impact on the GI microbiome in the infant. And studies have found that in breastfed infants, there's a decreased quantity of these bacteriophages, and that there are fewer pathogens, so fewer that are sort of detrimental. And human milk also contains viruses that are transmitted from the mother to the child via breastfeeding. So not bacteriophages, but some of the more typical viruses that we think of, things in the family of the papillomaviruses and herpes viruses, among others. We also see that there's a mycobiome. So there's fungus in human milk. A 2020 study showed that there were fungus was found in 20% of samples of human milk. There are a variety of different kinds, including Candida and some others that people may or may not be familiar with. And a study looked at the different types of fungus seen at different points in breastfeeding, and showed that there is a variable composition of the milk microbiome with maturity. So in the early milk, like colostrum, there's more of the Saccharomyces and Aspergillus compounds. In mature milk, penicillin compounds were most common, and some others that were seen earlier had disappeared. And there are also changes in the makeup of the microbiome throughout weaning. So we see that during the duration of breastfeeding and how far we are into the breastfeeding course can impact the microbiome. And then also one particular kind of fungus seen is Malassezia globose, It's seen in all healthy human milk samples, but it is not found at all in any that have been associated with mastitis. So this shows this interplay between the different parts of the microbiome. And this is an example of mastitis, which is a bacterial infection, can set up fungal dysbiosis. So it can impact sort of the balance of the fungus found in the breast milk microbiome. There are some other components, protozoa and archaea, that are also found in milk. So this is things like Giardia, Toxoplasma gondii. Another that is one that I can't say I was familiar with before this talk, that plays a role in weight regulation that other studies have found. So switching gears a little bit, but in addition to all of the microbes that are found in the microbiome, there are some other components of human milk. And I apologize because I think there's some window thing going on right outside my office. So I apologize if you can hear that. But there are prebiotics also found in human milk. And breastfeeding provides these human milk oligosaccharides that support the development of the infant gut microbiome. So interestingly, human milk oligosaccharides are the third most abundant solid component of breast milk after lactose and lipids. And they are these complex carbohydrates that are not digestible by humans. My window is being cleaned right now, so I apologize. The amount and the composition of these HMOs can vary among women, and also so differs within women and across the lactation period. So the total concentration of these HMOs is usually higher early in lactation and decreases within the first 3 months. And the content also varies depending on gestational age. So there are more of these HMOs after a term delivery than after preterm delivery. And formula companies have recognized how important these are in developing a healthy gut microbiome, and some infant formula is also fortified with some of these HMOs. So there's a lot of evidence to suggest that a lot of the health benefits of human milk can actually be related to these HMOs, the content and the particular content and composition in milk. These human milk oligosaccharides are metabolized by the microbiomes in the intestine. The metabolites are used as an energy source by the microbes and by the intestinal epithelial cells themselves, so the cells that sort of make up the lining of the intestine. And different human milk oligosaccharides have different properties and kind of assist in these processes through different mechanisms. So interestingly, there's a study that looked in these sort of organoids, these sort of like lab cultured intestinal cells, and showed that a complex human milk oligosaccharide that was taken from pooled human milk samples decreased a leaky gut in these organoids. And a leaky gut is one of the main factors that can worsen inflammation. And we'll talk a little bit more about that. So the HMOs were protective against this leaky gut and then inflammation. Studies have also shown that higher levels of specific human milk oligosaccharides in maternal milk are correlated with a decreased risk of some types of infections in breastfed infants. And right now, only certain human milk oligosaccharides are available at the industrial scale. So there are a lot of factors that affect the human milk microbiome. Some are environmental, genetic, immune related, and these vary both between and within individuals across time. There are prenatal, perinatal, and postnatal factors. And some of these are mode and timing of delivery, maternal diet and BMI, the use of peripartum antibiotics, mode of feeding, sex of the baby, parity, so birth order, and also geographic location. And this graphic sort of shows how this impacts child health outcomes. And so all of these different factors kind of coalesce to change the composition of the infant's microbiome. And as we've discussed a little bit, the composition of the microbiome is really important in sort of regulating how permeable the wall of the intestine is. So this gut permeability is a really important factor in the development of inflammation and later diseases. It impacts sort of how the immune system communicates across the wall of the intestine, and it can therefore kind of, depending if it's when it's not working properly, can cause low-grade chronic inflammation, and that can lead to later development of various diseases. And there's both the genetic predisposition that plays into this, and then also changes in the microbiome. So one thing that's been looked at is sort of the mode and timing of delivery. So studies have shown that labor might influence the human milk microbiome by increasing the intestinal permeability, and kind of ramping up that enteromammary pathway that we talked about earlier. So if labor allows for more bacteria and other things to be able to cross through the maternal intestinal wall, that allows more to sort of get into the human milk that's presented to the infant. So some studies have looked at mode of delivery, so C-section versus vaginal delivery, and found that C-section, the milk from C-section deliveries had a higher total bacterial concentration than in vaginal delivery, and more of certain types of species like strep, for example, which is common on the skin, and less of the bifidobacterium. And this may be associated with increased risk of immune-mediated and inflammatory disorders that some studies have shown are more common in infants born by C-section. Other studies show that C-section deliveries, the milk had higher levels of proteobacteria and then lower levels of Firmicutes, which includes lactobacillus. Interestingly, emergency C-section milk samples look more similar to that to milk from vaginal deliveries, because in emergency C-sections, typically people have had some amount of labor before they go to a C-section, versus in non-emergency C-sections where there's been no labor, those microbial profiles look more similar to that of the skin and the oral microbe communities. So that labor, at least having some labor, even if the baby doesn't pass through the birth canal, seems like it does have an impact. Not all studies have found these associations. And interestingly, at least in one study, the differences in the infant gut microbiome that were noted by mode of delivery were transient. And by 1 month, the study couldn't separate out whether infants had been born via a vaginal or a C-section delivery based on the components of their microbiome. There are also changes based on timing of delivery. So there are lower levels of certain bacteria in preterm deliveries than in the term group, and those changes stayed consistent throughout stages of lactation. So we're still, with the microbiome, we're still sort of too early to say exactly how true all of this is. There's definitely going to need to be more studies that look at mode and timing of delivery and how this impacts the microbiome. Other things that have been looked at are the maternal diet and also BMI, the maternal BMI. So in mothers with obesity, which is typically defined as certain BMI cutoffs, colostrum has more of lactobacillus as compared to mothers in the non-obese group. Their mature milk also had more staph and other types of bacteria, and then less of Bacteroides and the category that includes lactobacillus. Also, even separate from BMI is just looking at maternal diet. And the way that this sort of impacts the child is the maternal diet impacts the maternal gut microbiome. And then this, because of these pathways we've talked about, including the enteromammary pathway, impacts the human milk microbiome. And then that, through vertical transmission to the child, can then impact the child's microbiome. And so some specific type of nutrient-related changes in the microbiome that have been seen are listed here. So vitamin C has been associated with an increase in staph bacteria in the microbiome. Polyunsaturated fatty acids and linoleic acid can cause an increase in bifidobacterium. B vitamins are also found to influence the breast milk microbiome. and high intake of calorie-rich diets in pregnant women can lead to an increase in the Firmicutes category. So again, lots of descriptions, lots of associations, unclear exactly what this means downstream. So another one that gets looked at frequently is antibiotics. So some studies show that antibiotic therapy can alter the balance of microbiomes, and specifically some studies have shown a reduction in the bifidobacterium and lactobacillus species. One study recently found that bifidobacterium was present only in milk samples of mothers without the use of intrapartum antibiotics. And this reduction of bifidobacterium in some studies has been associated with a higher risk of allergic type diseases and also obesity. There are a lot of studies that have looked at mode of feeding in terms of breast milk versus formula, and found that there are differences in the microbiome when the stool is analyzed between infants who are fed human milk versus formula-fed infants. There's also starting to be some work looking at whether there's a difference in nursing, like at the breast, versus pumping and bottle feeding. Direct breastfeeding tends to impact or cause more of the infant mouth flora to enter into the infant microbiome, versus indirect breastfeeding may increase the acquisition of more environmental bacteria, because obviously there are more parts involved and more steps to get from the milk from the breast into the baby's mouth. The one study, there's a large cohort called the child cohort study that looks at over 1,200 infants and their mothers. And a 2020 study from this cohort looked at breast milk feeding practices and asked the question, does the mode of feeding matter? Does it matter if you nurse versus pump and offer bottles? And first of all, they looked at, does bacteria from the breast milk actually transfer to the infant gut? And does the mode matter, whether it's direct nursing versus pumping and bottle feeding? Interestingly, they found that there was no overall difference in the sort of diversity or maturity of the infant gut microbiome at 3 months based on the mode of feeding human milk, whether they nursed at the breast or pumped and took bottles. The sort of diversity and maturity of the infant's microbiome was the same. They did find that certain bacteria co-occurred in mother's milk and their infant stool. So this was sort of suggestive of the idea that human milk bacteria does transfer from the mother's milk into the infant's microbiome. And they did find that this co-occurrence was reduced when the infant was receiving pumped breast milk as compared to directly nursing at the breast. They also looked at sort of the relative amounts of some of the common species between the mother's microbiome and the breast milk microbiome versus the infant's gut, and found that there were sort of similarities between the breast milk and the stool of the infant. So more evidence that the microbes do transfer from breast milk into the infant gut. And in this study, they found that the composition of the infant gut microbiome was strongly associated with breastfeeding, like with exclusive breastfeeding and the duration of exclusive breastfeeding, but not so much the mode of feeding, so nursing versus pumping. So it seemed that what was really impactful was how long infants received exclusive breast milk and not so much how they received it. Interestingly, though, this same cohort, there are lots of studies that have been done on this cohort, and the authors of this paper referred to another study of the same cohort that showed slightly weaker protection against asthma and being overweight in children who had actually directly breastfed versus taking pumped milk. So there's obviously still a lot of research needed to be done to sort through this. This study looked at the use of exclusive human milk versus bovine milks in preterm infants under 30 weeks. So some studies have shown that there is a decrease in morbidity among these early preemies when they're exposed to exclusive human milk rather than getting formula and other milk fortifiers that are made from bovine products. And so this was actually a randomized control trial. A lot of the studies that I've discussed before and that have looked at the microbiome have been sort of cohort studies, and this a randomized control trial is sort of the gold standard in terms of trying to sort through causation rather than just associations. And this looked at exclusive human milk in these preemies less than 30 weeks up till they were 34 weeks post-menstrual age. The experimental group received only pasteurized donor human milk and human milk derived fortifiers, and then the control group received infant formula that was bovine, from a bovine source. And this study did not find any difference in the bacterial diversity between the two groups and didn't find any difference in other outcomes like time to full feeds, risk of necrotizing enterocolitis, or other morbidities. So this doesn't fully answer the question for sure. It's just one study, and it was about 120 infant mother dyads, but this particular study did not find a difference in the microbiome of these infants receiving only human milk versus bovine products. So other factors that have been looked at in terms of different composition of infants' microbiome and of human milk microbiome. So baby sex, because the oral microbiome of male versus female infants leads to differences in the retrograde flow into the breast and then into the milk. Maternal parity, so birth order, staph and Haemophilus are more abundant in bacterial samples of moms that have had other children than in those who are having their first child. Maternal psychosocial distress is associated with lower bacterial diversity in human milk microbiome 3 months postpartum. And geography, so in rural areas, there's more bacterial diversity typically in the human milk microbiome compared to urban areas. So why does this matter? And this is sort of getting towards our third objective of how does this impact outcomes in children? And this is just sort of one snapshot, but to sort of summarize, infants who primarily breastfeed during the first months of life share 28% of their stool microbes with their mother's milk. So the mother's milk has a large impact on the baby's microbiome. And the microbes in the milk help regulate the immune function in the infant. And so how does this all play out? And so this is a graphic that sort of just puts this all together. We see here these perturbation risk factors. So basically all the factors that we just talked about that might have an impact on the human milk microbiome, which contains the different factors, bacteria, viruses, fungi, archaea. And this human milk microbiome is key in sort of creating the gut microbiome of the infant, and also through this gut respiratory access, the respiratory microbiome. And so the human milk microbiome, along with the gut and respiratory microbiome, can sort of go in two different directions. We can have a healthy microbiome that is prone towards being able to be resilient after a stressor, or we can have dysbiosis, where after a stressor, the microbiome can't recover. And this is where we sometimes start seeing diseases like neck and asthma and obesity later in childhood. So we're moving on to our third objective. So the role of the human milk microbiome in health and disease. So we've talked a lot about this, but there's sort of two parts to it. One is to promote the intestinal immune homeostasis. And the other is to sort of just more directly facilitate digestion in the infant. And in terms of this intestinal immune homeostasis, there are lots of parts to this. For one thing, bacteria from the human milk microbiome are sort of the first type of bacteria, or one of the early types of bacteria that the infant comes into contact with. The infant gets antigen stimulation for their intestinal immune system and starts to sort of learn balanced responses to being in contact with the bacteria. These microbes from the human milk microbiome that then enter into the infant also help with fermenting the human milk oligosaccharides, like we talked about, these produce different products, short chain fatty acids that have an impact on sort of the host immune system and those processes. And some of the bacteria, such as lactobacillus, also help promote a balanced immune system by kind of getting in the way and taking up space and not allowing the proliferation of other sort of bad actors. And so this leads us to thinking more about this sort of resilience versus dysbiosis of the human milk microbiome. So as we've said, the human milk microbiome changes with stress, and following a challenge, the microbiome of a healthy individual is able to return to baseline. If not, then a new equilibrium is established that sort of tends towards dysbiosis. So resilience is a marker of a healthy ecosystem. The microbiome of a healthy person can resist change under stress and can kind of fully recover after some kind of perturbation, whether it's an illness or an antibiotic or something else. And the human milk microbiome is a fundamental contributor to the gastrointestinal colonization in children. So children need a healthy intestinal bacterial colonization to prevent and correct for dysbiosis to minimize the impact on later health. And dysbiosis, we can, as we will see momentarily, can be quite dangerous. So this just kind of is a graphic that explains a little bit of what I've been talking about of this, the importance of the gut and the proper functioning of the cells that line the gut, and why this matters in terms of dysbiosis and inflammation. And so in a normal, healthy gut, we see these are enterocytes. These are the cells that make up the lining of the intestinal wall. And this side here is where food and other things come through, and our body has to figure out how to not allow everything that comes into our gut to actually get into our bloodstream and impact our immune system and development of allergies and things like that. And as the health of the gut sort of decreases, we get more leaky junctions here, like more can pass through here, and eventually we can get into a system of like significant dysregulation, and lots can come through. And so the more that sort of inappropriate products come through the intestinal wall and end up getting into our bloodstream, the more we get into this sort of pro-inflammatory sort of allergic state. And the microbiome has a large role in promoting the health on this side and helping with mucus production that is an extra barrier, and other compounds that kind of help keep the gut in good working order. And so one example of this is of where this can go wrong in a very direct way is necrotizing enterocolitis. And so for many of you have probably heard of this, but some may have not. This is an intestinal infection that is seen at times in preemies. It's where the intestinal lining becomes inflamed and then can die, necrose, and eventually perforate. And this can lead to sepsis, so a bacterial infection in the bloodstream, peritonitis, so a bacterial infection of the sort of contents of the abdomen, and at times death. And it can affect 5 to 10% of preemies under 1,500 grams, and has a very high risk of morbidity, like short bowel syndrome and liver failure, and also mortality, which can be seen in up to 50% of infants who end up having NEC. So it's a very serious and concerning issue. The etiology of NEC is not totally clear. It's likely multifactorial, and it doesn't seem to be related to any particular bacteria. But many studies have shown that there's a lower incidence of NEC in human milk-fed babies as compared to those who are receiving formula. And there's an increased incidence of NEC when babies are exposed to antibiotics and proton pump inhibitors, so like antacids, like omeprazole, because both of these are things that modulate the microbiome. They have an impact on the microbiome. And so in preemies, there's this sort of hyper-inflammatory response to dysbiosis that happens in the intestine, and this can lead to NEC. And some factors with this are that the preemies, their intestine is immature, and when they're born early, they're at a stage where they're used to sort of only seeing a few microbes in the intrauterine environment, and instead they're exposed to all of these colonizing bacteria because their microbiome sort of ramps up more quickly, and they therefore don't have the normal pathways in place, and things are sort of out of whack, and this leads to excessive inflammation. And then this ends up leading to tissue damage and loss of the intestinal barrier integrity, and sort of what we saw in that last slide. And so here is a healthy, normal intestinal wall where we see these tight junctions, not much can go through. We see this commensal bacteria, the yellow rods here. This is the microbiome doing its job in part by actually being a barrier. Also, the microbiome helps with the production of this mucus that sits along the wall of the intestine, and IgA, which is the primary immunoglobulin that comes through breast milk, is also active here. And so this is part of what helps keep the normal lining of the gut working properly. And what we see in necrotizing enterocolitis in these preemies is that, one, they may have been exposed to antibiotics and acid reducing agents, possibly bovine milk related formula. And they have this sort of immature intestinal barrier in the first place, and an immature immune system. And all of this comes together to create tissue injury and intestinal necrosis. And some of these normal processes, like the microbiome, which is sort of not set up properly yet, isn't here. There isn't as much of that mucus, and not as much of the immunoglobulin that typically comes through breast milk. And so there's a lot of work looking at NEC and dysbiosis. And so this study from 2016 was a cross-sectional birth cohort study, and it found a sort of signature microbiome of very preterm infants who eventually went on to develop NEC. And so there were positive associations with certain types of bacteria, and then negative associations with other types of bacteria. And so basically the microbiome of infants who developed NEC versus those who did not were different, and in some consistent ways. And so the hypothesis is that atypical oral intake, like delayed enteral feeding, when some preemies may not be able to even start using their gut initially and are being fed through or given nutrients through an IV. They're getting tube feeding, where first of all, there's a piece of plastic sitting inside of them. They may be supplemented with formula. Also, ubiquitous exposure to antibiotics, because when babies come early, almost 100% of the time, they're put on antibiotics for a question of whether infection was the cause of their prematurity. And all of this can play a role in sort of changing the composition of the microbiome. And as I've said before, feeding micropreemies, mostly human milk, does seem to decrease the risk of necrotizing enterocolitis. Some of the possible mechanisms are that the maternal microbes are transferred via human milk. Human milk contains these human milk oligosaccharides, other immune mediators, and then a complex interaction of all these factors. And then other factors like exposure from NICU, C-sections, limited breastfeeding, less skin to skin can also lead to dysbiosis and a pro-inflammatory microbiome. And so this slide sort of goes through some of what human milk provides. There's other studies looking at this sort of gut-lung axis, and that the GI and the respiratory tract dysbiosis can impact development of respiratory diseases such as asthma. The main source of lower airway colonization in the microbiome is the upper airway, and also some impact from the circulating compounds from the gut microbiome. And so early, healthy, sort of diverse, non-pathogenic colonization of the lungs may be protective the gut may be protective for the lung microbiota. And so there's some studies that show that differences in the microbiome of the healthy respiratory tract that there are differences in the microbiome of a healthy respiratory tract versus the microbiome of children with asthma. And interestingly, a study found that C. diff colonization at 1 month correlates with wheezing and asthma up to at age 6. So some really downstream impacts from the differences in the microbiome. A 2021 longitudinal study showed that children at risk of asthma had transient intestinal microbial dysbiosis at some point during the first 100 days of life, and that this set them up for asthma later. Also, studies have shown that the maternal diet, so a diet that's prone towards sort of high fat diet during pregnancy, as I mentioned earlier, can contribute to lower levels of Bacteroides in the infant gut, and that this may be a predisposing factor for later developing asthma. A lot of work looking at obesity in children, and this is definitely an area for further research. A lot of longitudinal studies that are looking at the microbiome of children who later develop obesity versus those who don't. It's been established that breastfed infants have a lower incidence of obesity than formula-fed infants. And the question is this because of the microbiome, and what other factors may contribute? A recent study showed that mothers with obesity provided their infants with less breast milk at 6 and 12 months compared to quote unquote healthy weight mothers. And a shorter breastfeeding period is associated with more minor diversity in the gut microbiota of children. And so is it possible that this is part of the pathway that leads to increased risk of obesity in non-breastfed infants? There are other things that are being looked at. It's likely that the microbiome impacts a multitude of conditions, different atopic diseases other than asthma, so food allergies and eczema, different gastrointestinal diseases, IBS, so irritable bowel syndrome, autoimmune issues like celiac and type 1 diabetes, a lot of work on neuropsychiatric diseases like autism, anxiety, and depression, and even cancer. So I'll just very quickly go through this because I know we're mostly out of time, but there are some factors that modulate the microbiome that are sort of being studied to figure out what can we do about this. So for one thing, pasteurized donor human milk, how can this be personalized? So mom's own milk, that's the MOM, is the gold standard for feeding preemies, but typically quantities are insufficient. And so we end up supplementing often with pasteurized donor human milk. The issue is that the pasteurization kills the microorganism. So it gets rid of a lot of the microbes that we've been talking about. So the preemie GI microbiome of infants who are fed mother's own milk was different than that of preemies who are fed pasteurized donor milk. Something is missing. And so this study looked at how can we personalize the donor milk by inoculating it with small quantities of mother's own milk. And so this 2017 study, they tried different methods. And what they found was that if they were able to use at least 10% of mother's own milk, incubated for 4 hours at 37 degrees, that they were able to partially restore some of the microbiome and microbes that we typically see in mother's milk. This has the effects on the infant GI microbiome and infant health have not yet been studied. This still didn't restore other components of the breast milk, like immunoglobulins and some other proteins that are degraded by even this method that was used. And there's work looking at whether UVC treatment or other types of pasteurization might be preferable to the Holder pasteurization techniques that's used now that might retain more of these other components. And then infant formula, lots going on to understand more about what's in breast milk that makes it so helpful to the infant microbiome. So we've talked about human milk. Oligosaccharides are also are already being added to formula. And then there's other particular bacteria that may be sort of like a pioneering species that needs to be there first to help set up a healthy microbiome. And so questions looking at whether we can try to replicate that for formula. And then lots going on also to look at the role of probiotics. So this was American Academy of Family Physician review on the role of probiotics in necrotizing enterocolitis. And this overall found that use of probiotics did reduce the risk of severe NEC and mortality in preemies, and that there was a reduction in incidence in the mortality of NEC when probiotics were used. Of note, the main medical center, NICU, does not routinely use probiotics, and part of the difficulty is that probiotics are they're not, they're considered supplements, so they're not regulated in the same way that medications are. And I think that's part of why the MMC NICU doesn't use probiotics, because there isn't one that's sort of FDA approved, and that's part of the barrier. So just to define probiotics, they're live microorganisms that, when administered in adequate amounts, can confer a health benefit to the host. And there are lots of ideas of how this may be. Lots of people are taking probiotics for all different reasons. There's not a ton of evidence yet. The one place where probiotics are, there is evidence to show that they're helpful, is for breastfed infants who are experiencing colic. There are a number of studies that have found that probiotics can, and in particular, a lactobacillus compound, can actually be helpful for decreasing colic in breastfed infants. Everything else, lots of questions. So for example, probiotics for like frequent spitting up. Some studies show that it might help, but there is not enough evidence to suggest that. And this, I'm not going to go into detail, but just all these different conditions where probiotics have been examined. So in summary, the microbiome is hugely important in concert with genetics in terms of impacting human health. It's diverse and dynamic. Human milk is a key factor in shaping children's microbiome. It plays a major role in shaping health and disease, and there's definitely much more to learn. Many areas of research are needed, many opportunities for education and advocacy. We're at a stage where there are many observations, some associations, and not necessarily a ton of causation yet. Lots of future directions, and some are already here in terms of some of what is going on in terms of probiotics and other medications that are trying to kind of impact the leaky gut and things like that. So I am happy to answer questions if people are able to stay for another minute or two, and my email is there also if anyone wants to email me with any questions.
Wow, that was fantastic, Jess. So much really rich information process. So
I know it's a lot.
yeah, it's really, really fascinating. And I'm so glad that this research is being done to really better understand how this all works, and to continue to make the argument that breast milk is really powerful and really important to continue to protect it. Any questions from others? You're welcome to unmute or type in the chat box. So we can send a certificate of attendance if you're interested. We don't have CEUs for this, but we can send a certificate of attendance. If you want to send a message to me at mainbreastfeeds@gmail.com, I'm happy to put one together and send that to you. And Jess, will you make this PowerPoint available to share as well? Is that possible?
Yes. Yes.
And the recording will be on our YouTube page within the next 24 hours or so. And I'll send that out in our email too, once that is up. I know I'm personally thinking a lot about my firstborn, who was born 11.5 years ago, and the 7 rounds of antibiotics he received in his first year of life due to ear infections. And he has asthma and food allergies, environmental allergies, and eczema. He's got all of that. And he was also exclusively human milk fed. And I think about the dysbiosis that would have been happening had he not been able to get that human milk, if his conditions would be worse. Yeah, there's just so much. I have some other questions, but I know that we're out of time essentially, but let me just see. I think Mary has a question. If I understand correctly, any amount of mother's milk is helpful from a microbiome standpoint. When we have moms using donor milk or moms who are working on their supply, and in the meantime, babies needing to be supplemented, providing information that their milk is still super important. Yeah, I think I see.
Completely agree.
Thank you.
And I think one reason I added advocacy, the mention of advocacy, is that there is, I worry that some of this feels like it's a lot of responsibility for mothers, and people, there's many ways to sort of promote your child's health, and it's sort of not there's not just one way to do this, but these are just important factors to understand. And also, then we really need to kind of go up the chain of command. And so be able to support people so that they can do their best.
Exactly. Yeah. And in your PowerPoint, yeah, all of as many references as you can include would be fantastic. Jess, thank you. Kylie, yeah, I see your question. I don't know did she just drop off? Oh, no,
I think
Kylie.
she's still there.
Okay, would you like to unmute and ask your question? I think it's a great question.
Yeah, sure. This was just chock full of information, so I so appreciate it. I was taking notes, but I'm curious if any hospitals are doing that recolonization of PDHM with the 10% of mom's own milk, and if so, how they're incorporating that in their workflows.
I don't think that my sense is that's not actually being done yet.
Yep.
It looked like I looked at that, and then also the UVC pasteurization, and it looks like that is sort of like an active area of research, but I don't think anything that's actually in practice yet.
Gotcha. Lots of potential here.
Mhm.
Fascinating. Thank you so much. Thank you everyone for joining, and we will stay in touch. Have a great rest of your day.
Thank you, everyone.